Good evening, ma'am.
Good evening, sir.
Good evening, Gana. Are you all right now? Hello?
No, sir, still fever is there, sir.
Can you attend now?
Yes, sir. Yes, sir.
Okay. Finished within 40.
Good evening, sir.
Good evening, ma'am. Good evening, ma'am.
You can start, sir.
Yes, ma'am. Yes, ma'am. Sir. Thank you. So, good evening, Ajay. So, today we are going to discuss. So, what are the discussion already we did you know? So, already know a little bit. What is the main structure of the sperm? Particularly, what are the parts is present in it? Already you know a little bit. And then, so what is by fertilization, otherwise called as syngamy? That things also already we discussed, isn't it? Male as well as the female gamete united together form as a diploid zygote. That things already we discussed, isn't it? After the fertilization, what are the events which occurs? Okay, that things we are going to discuss here. What are the things is that now there are so many things already we discussed across since so many enzymes also involved in this process to penetrate the sperm into the egg. That things also already we discussed, isn't it? Okay. Today, what we are going to discuss, see the specific. So, see here, the species specificity in fertilizations. Already you know about what is meant by fertilization. Just one. Okay. Anyway, species specificity in fertilizations. Here, yeah. Okay. The egg, otherwise called as ovum, secrete a chemical named, is called as fertilizing. Once again, repeat for you. Egg, the secretor secretes a chemical name, is called as fertilizing. It is composed of the glycoprotein, and it's nothing but the protein molecule, isn't it? Glycoprotein. Divide into two things. Glyco is denoted as monosaccharide, otherwise called as glycosidic. What is the meaning of glycosidic? Means the bond between the sugar molecule, isn't it? So likewise here, which saccharide present in monosaccharide? Okay, the monosaccharides here. Protein already in our but amino acids. What I am telling fertilization is nothing but the glycoprotein. It is composed of the two components, glycoprotein. Glyco means sugar molecule, as well as amino acids is nothing but the protein molecules. These two combination is called as fertilization. Okay. Right. The sperm has on its surface protein. Not only that, and also the sperm also it has a surface protein substances called anti-fertilizins. Okay, here fertilizin, here anti-fertilizins. So what about this composition? She is composed of the acetic amino acids. Acetic means COOH, carboxylic manner. Okay, so these are the two things is the first happen. Once again, specificity of the fertilization. So particularly the egg is very, very important. They secrete the chemical name. It is called as fertilizin. It composed of the glycoprotein. The glycoprotein, what are they? Monosaccharide and also amino acids. And second thing, what is that? Sperm has on its surface protein substances called as anti-fertilizin. Fertilizin, anti-fertilizin, already told. It is composed of the acetic amino acids. Acetic amino acids. There are so many things. Okay, these are the 2 points. And then the fertilizin of an egg interact with the anti-fertilizin. These two are react together. Of a sperm of the same species, this interaction make the sperm sticky to the egg surface. Sticks mean adhere on the egg. So these are the two protein components. What is that? So react together. Okay, interact together with the egg layer, particularly egg layer. So as a result, it sticks to the egg surface. It can adhere on the surface of the. So sometimes they may ask the question. Okay, the sperm. Okay, the sticky to the egg surface. Which one of the following component? One is fertilizin or anti-fertilizin. Sometimes they ask the individually fertilizin only, anti-fertilizin only, anti-fertilizers and fertilizin only. Like that, they may ask the questions. Okay, so you have to answer it. So what is the main function of these one? These two are react together. It is main function of the sperm as well as the egg adhere on the surface. That is the main reason. Okay, the next one. See here. The adhesion of the sperm to the egg of the same species through the chemical recognition is known as agglutination. Once again, repeat again. It's only adhere on the surface, only the same species only. For example, mammals, take an example, so monkey, rabbit, okay, rat, and chimpanzee, human beings. These are the same species, isn't it? Homo sapiens. But at the same time, when you are taking the another species, it will not easily recognize the chemical substances. Chemical substances already we discussed. Which one you know? That fertilizing, anti-fertilizing is a component, isn't it? That component is considered as a chemical substance. It's specifically secreted by the particular species. That's why they have given us a title is what is the specificity of the fertilization. Okay, so this is called as an agglutination. When the egg and sperm chemically united, recognized, that process is called as agglutination. That is clumping. Okay, so this is very, very important. Next, what is the main significance of the fertilization? What is the main significance? Here, see here. In fertilization process, a number of chromosomes are deployed inside, isn't it? Because fertilization means what is a male gamete as well as a female gamete united together. A female gamete is it has haploid. Okay, male gamete is also it has haploid nature, 23 chromosome. Both are 23, 23, isn't it? They are all N and N. United together, you will get diploid nature. Diploid means about 2N stage, that is 23 plus 23, you will get 46. That is called a zygote, isn't it? That is the first significant point. Second, during fertilization, listen carefully. During fertilization, male and female chromosomes are combined. That is cross-pollination. Already we studied where will already we studied meiotic cell division, isn't it? It is occurs in the reproductive cells. So the male and female chromosomes are combined and crossing over takes place. that lead to the variation offspring. Already we studied, isn't it? Because we are all our offspring. Okay, we are the combination of both mother as well as the father chromosome. Okay, isn't it? So we are the varied offspring always. Which one you know? Variation. Variation means what is that? That is our offspring because both the character present. That is the reason. Okay. In fertilization, genetic variation. What is the genetic variation? The chromosome of from the 23 from male gamete, from 23 from female. The genetic combination united together as a result, the crossing over takes place, isn't it? Crossing over taking place. As a result, what happened? You know, the variations occurs. Okay. So the variation is essential for the evolution, they told. The in fertilization, genetic variation, combination of both the male and female, isn't it? Develops, so it is essential for evolution. Evolution means it adhere or inherited from one generation to another generation. That is very, very, very important. These are the significance. First, what happened? Diploid zygote formation. Then what happened? Crossing over takes place. As a result, what happened? Evolution takes place. Okay, got it? Okay. So go to the next one. So, types of egg membrane. Suppose taken examples, a egg. When we concern about the egg, it has a layer, isn't it? How many layers are there? Anybody else? Agil, Jaswant, and Ajay. If you can, please go ahead. If you can. Okay, I will tell you. No need to risk. Okay. Types of egg membranes. Here, there are 3 types of egg membrane have been reported. But taken any one of the egg or duck, or taken examples of hen, whatever it may be, in human beings also. There are 3 types of membrane. They are basically divided. One is called as primary membrane and secondary membrane. Third one is called as tertiary membrane. The primary membrane, which one is secreted? You know, see here, the primary membrane is the outer layer, isn't it? Secondary membrane is also another layer. Tertiary means third layer. Total, how many layers present? 3 layers are present. Okay. The primary membrane secreted by ovum. Primary membrane secreted by ovum. Example, vitelline membrane. So vitelline membrane is a primary membrane. Zona pellucida of human egg is also a primary membrane. Both are secreted by ovum. Okay, primary membrane, vitelline membrane, as well as the zona pellucida is an example. These are secreted by ovum. Okay, so secondary membrane. The secondary membrane was secreted by ovarian follicular cells. Here ovum, here ovarian follicular cells. Example, corona radiata of human egg. So only one is been reported here. The secondary membrane is corona radiata of human egg. Third one is tertiary membrane. Tertiary membrane secreted by oviduct. Here ovum, ovarian follicle, and oviduct. Okay. Example, albumin cell membrane and cell of the egg of the reptiles and bats, jelly layer in egg of the frog. These are all are come to the conclusion. Tertiary membrane. This was secreted by oviduct. All of you, please kindly take a hints. Take a note or paper. Just note it down. Types of egg. Put a heading. Primary membrane secreted by ovum. Example. Secondary membrane secreted by ovarian follicular cell. Example. Tertiary membrane, oviduct. So what are the examples? You have to note it down. That is very, very important. Please follow it. Okay. Go to the next one. See that already we discussed this thing. There are 3 types of egg membrane. The primary egg membrane formed by the egg itself, side of ovary, zona pellucida, mammals of chorion in insects. Okay, chorion is insect. Secondary egg membranes formed by the ovarian tissues. Okay, before the egg leave the ovary. Tertiary egg membrane formed during the passage of egg in the oviduct. Example, egg of the toad or egg of the bats. These are all the 3 types of egg membrane. Sometimes they may ask the question in the match the following questions. Okay, for the next one. See here. Once again, this is the egg. Okay, this is the egg. See, what are the layer present in it? See here. The zona pellucida. Okay, zona pellucida. See here, this is called as zona pellucida. This is the layer that is called as zona pellucida. It is a thick membrane. Exists for sometimes post-fertilization. An outer protective layer. This is the outer protective layer. Which one is called? This one is called as outer protective layer. Okay, this one is the inner one. Okay, this one is the outer one. Okay, and second one is called as corona radiata. Second one. These are the structures. Egg. Okay, corona radiata. The above the which one zona pellucida, there is a outermost layer. Okay, see here. 2 to 3 layer of the cell attached to the zona pellucida. See here, this is the zona pellucida. A 3 to 4 layers are surrounded. 3 to 4 layer surrounded. That is called as corona radiata. Okay. Supply vital protein to cell. It produce the what happened? It provide the nutrition in the form of proteins. Okay, that is very, very important. See here. So we come to the point, outermost to inner. So outermost layer is called as corona radiata. The middle portion is called as zona pellucida. And third one is called as vitelline. Say that vitelline membrane. So outermost to inner, that is your better choice. C jet V. C jet V. C denoted as corona radiata. Jet denoted as zona pellucida. And V denoted as vitelline. Okay. The vitelline membrane, the plasma membrane of the egg cell. See here, this is the egg cell. That is the outer covering is called as what is that? Vitelline membrane. Control entry or exist of substances. Substances means what is that? There may be protein or nutrients or whatever it may be. Vitelline membrane, it acts as a semi-permeable membrane. Semi-permeable mean what is the meaning? Selectively permeable. Sorry, semi-permeable or selectively permeable. Selectively permeable. Okay, it will not allow all the components. It will not allow all the components. Which one they need? Then that content only they will be move inside with the presence of vitelline membrane. And the next one is called ooplasm. Ooplasm, what is the meaning of ooplasm? Means is near similar to that of the cytoplasm of the egg. This is the egg. So this is called as outer. That is called as ooplasm. Vitelline layer. The innermost egg layer, in between there is a space. The space is filled with the ooplasm. Okay. Cytoplasm of the egg cell. Sometimes they may ask the question. Cytoplasm of the egg cell is also called as ooplasm. Supply nutrients to growing embryo. What is the main functions of this ooplasm? They provide the nutrition to the growing egg embryo. That is very, very important. After the fertilization, it is provide the protein molecule. Which one? Already you know about it, isn't it? Corona radiata provide the nutrition for the things. Vital membrane is also control the substances. Inner side there is a cytoplasm, like as a cytoplasm, that is called as ooplasm. The ooplasm contain nutritive substances for the growth of the embryo. That is very, very important. Okay, for the next one, the germinal vesicles. See that the inner side there is a one layer is that is called as germinal vesicle. Okay, the germinal vesicles nucleus of the egg cell and contain genetic materials. Note it down. This is very, very important. Nucleus of the egg cell. This is considered as a nucleus of the egg cell. It contains what is that? Genetic material. What is the genetic material? Already know very about DNA as a genetic material. isn't it? DNA present in what DNA? Both male as well as the female DNA molecule present inside, isn't it? Next one is germinal spot, nucleus of the egg cell. The center, there is a germinal spot is there, that is considered as a embryo developing portion. Okay, so these are the entire structure of the, which one you know? Ovum, zona pellucida, corona radiata. You start from the corona radiata because it's outermost layer. The next one is called as, what is that? Zona pellucida, vitelline membrane, ooplasm, and also germinal vesicle, and also germinal spot. Like that, you have to keep it in your mind. Okay. Okay. So go to the next one. So the egg membrane already discussed, plasma membrane, vitelline membrane, and secondary. Sometimes they may ask the question, which one is considered? It is primary egg membrane, primary plasma lemma. Okay, vitelline membrane. These are all are what is that? Primary egg membrane. Secondary egg membrane. See here, zona pellucida, corona radiata. These are all are considered getting known as secondary membrane. Okay. Likewise, you have to keep it in your mind. Okay. See the female egg structures: corona radiata, follicle cells, cytoplasm, nucleus, plasma membrane, and also zona pellucida. These are the another structures. Okay. Likewise, I have selected so many slides. Okay, slides for your reference only. This is oocyte membrane. Oocyte membrane inside the membrane. Okay, so here zona pellucida, so corona radiata. The completion of meiosis to the sperm is entered. Already I have sent one video also, AV also. Okay, just refer it, you will get the ideas. Polyspermy block, because once what happened in oocyte activation trigger the polyspermy. Polyspermy means, what is that meaning? More than one sperm reach the egg, isn't it? So it will not allow. It will not allow block of polyspermy. So after the completion of the meiosis 2, which one? The oocyte membrane. Okay, this is also a very, very important. Okay. So this is also total egg. Sometimes they ask the question, when we are consuming the normal egg, you know, hen egg, the outermost layer is called as, what is that? Egg cell, isn't it? The egg cell is made up of calcium carbonate. Our teeth is made up of calcium phosphate. Okay. Then outer membrane. Above the skull, cell, the inside there is a two layer of the membrane. One is called as outer membrane and another one is called as inner membrane. Outer means, you know, it may be faced toward the periphery. Inside there is a inner membrane. So this portion is called as chalasa. Chalasa means just to go to the, which one you know? Flower part. Chalasa, when you are going to take in ovary structure in plant, the chalasa part is there, isn't it? Okay. Exterior albumin, outside the albumin position. See here, outer thin albumin. Thin albumin, it is a very light, small amount only present. That one is called as outer thin albumin. Inner there is a thick albumin. Thick albumin means its compound will be very high. Okay. And center there is a vitelline membrane. See a nucleus panther. This is called as nucleus panther. And germinal disc. This is called as germinal disc. And the yellow yolk. Yellow yolk is also a covered white yolk. So both are present inside the germinal disc, inside the blastomes. Internal albumin is also present in this area. There is an internal albumin. This one is called as external, isn't it? The external albumin is outside. The internal albumin near to the, which one you know? Egg yolk. Chalasa, again chalasa is also, the both side there is a chalasa part is present. It's just like a twisted, what is that? Kite, whatever it may be. In the air cell, this position is called as air cell. Already we saw it, isn't it? When you are boiling the egg, there is a small gap is present in it. Okay, that is called as air cell. And also another one is called as cuticula. What is that exactly? We don't know. I do not know. What is my cuticula? There is a space. They don't denote this area only. Cuticula, like that they told it. So these are the egg structures. Why we are studying this one? You know how this one is, what is that? both male as well as the female part present inside. After the fertilization only you will get the egg, isn't it? In the lower category. And human being, what happened, after the fertilization? See, already we discussed these things also. Okay. So this is on the human egg. That one is called as hen, a duck, or whatever it may be, like that. Okay. We'll pass on to the next one. Is the gametes already discussed, isn't it? What are the types of gametes? What are the types of gametes already studied? There are 3 types of gametes already studied, isn't it? What are they? Isogametes, anisogametes, and oogametes. Where did you study it? Will you please? It is mostly found in fungi, isn't it? Isogamete, anisogamete, and also algae. Algae, the sexual reproduction mostly takes place by spores. Okay. And sexual reproduction, particularly spores, means they are having 3 types of spores, male as well as a female. Isogametes, listen here. Here, isogamete, in this male and female gametes or similar structure is called as isogamete. Iso means same structure. We cannot be able to differentiate which one is male, which one is female. That is called as what is the isogamete. Structure will be similar. Anisogametes means the male and female we can easily differentiate. Okay, is called as anisogamete. Oogametes is also what is that? Same. Okay, it's the largest one, male as well as the female, largest one. But female is also motile. Sometimes male is motile, like that. See that structure, you can understand. Okay, this is also very, very important, my dear students. Listen. First one is called as isogametes. See here, this is isogametes. Both are similar in nature. Which one is male, which one is female? We cannot be able to find out. Such type of category is called as what is that? Isogametes, mostly found in cladophora. Note it down. Isogametes, cladophora. Heterogametes. See here, these are the heterogametes. This is called as male gametes. This one is female gamete, because female gametes always large in nature. Already in our bird, isn't it? That's why. So this is the female. This one is called as male, because male gametes always minute in nature, microscopic in nature. So we can easily differentiate. That is called as what is that? Heterogametes. Where is found? Fucus. NEET question. Note it down. Diagrammatic question. They may ask the question. Sometimes direct question also, sometimes assertion reasoning also, sometimes it may ask which one, match the following. Note it down. This is very, very isogamete, cladophora. Heterogametes, fucus. And heterogametes of human beings, this is also a heterogametes. This is male, this one is female. Similar to that of the, which one, fucus. Here human being, this is a plant. This one animal, this one plant. Can you understand? Do you understand this one? Any doubt? Ajay?
Understood, sir. No doubt, sir.
Okay. See here. Same things also repeated here. This one is isogamete, heterogametes of these 2 types. Okay. And sex of the baby is decided during fertilization. After that, what will happen? The male and female are united together. As a result, the zygote formation takes place. After the zygote formation, what happened? Divisions taking place, isn't it? Then how the baby is after the fertilization, how the baby or whether the embryo is male or female, how they are decided? Because the presence of chromosomes already in our XX denoted as female, XY denoted as male, isn't it? That things has decide. The chromosomes pattern in human female is XX, and that in the male is XY. Here, what happened? Here the male, the X is also 50% present in it. Here both are same, isn't it? XX, but from the female there is a 50% of the character is here X. The Y is the deciding authority of the male sex. boy. Okay, think about the boy. The Y is a deciding characteristic. Otherwise, called as Y chromosome is the deciding authority of the male boy. Okay, fetus. Suppose XX means female. Okay, therefore, all the female ova gametes have the sex chromosome X, isn't it? All the female ova, it has a XX chromosome. Whereas the male gametes, sperms, the X chromosome could be either X or Y. So in this portion, 50% what will happen? Y chromosome is there. That is very, very important. After the fusion of the male and female gamete, the zygote would carry either XX or XY, depending on whether the sperm carrying X or Y. Take an example. Female always carrying which one? Which type of gametes? X gametes only, isn't it? X type of chromosome will be transferred. But in this category, which one male, the split gives two, what is that? Gametes, X as well as the Y. Suppose X combined with the X, you will get the female. Suppose X combined with the, here the X combined with the Y, you will get XY. So it is male. Likewise, what happened, deciding. Okay. So this is the important thing. See that the zygote carrying XX would develop into female baby, and the XY would form a male. That why scientifically it is correct to say that the sex of the baby is determined by the father and not by the mother. Note it down. This is very, very important. Okay. So here, why, here the both the sex are same, isn't it? XX are same. But here, X and Y. So the baby is determined, sex of the baby. X, here the father X is the, here the mother gamete is also. Both are united together, form as a which one, female. Suppose the Y is joined with the X, what happened, male. You got it? So, the sex of the baby is determined by the father and not by the mother. So that is very, very important. Reasoning, assessment reasoning time, straight questions. Sometimes they may ask the questions. Okay. See another one. Error in fertilization. During the fertilization, what happened? Always the fusion of gamete is not possible. Sometimes some error is also possible during the time of fertilization. What are the errors they may occur? One is called as polyspermy. Poly, already about what is by poly? More than one sperm fused in an ovum. Only one egg is there, but there are so many sperms also united. That condition is called as, what is that? Polyspermy. See here, egg is one, isn't it? See the surrounding, there is so many sperms is there. Suppose all are united together at a time, what happened? All are penetrated inside, means all the male gametes introduce the, which one, nucleus inside. That condition is called as, what is it? Polyspermy. Polyspermy, sperms, polysperms, more than one sperm. Okay, you got it down. This is also one of the error in fertilization. Sometimes it happen like that. It is not possible in rare cases. Okay, rare cases it is possible. Okay, in one girl they may get 6, what is that? Baby. That is also sometimes happen. All are 6 or 4 or 3. Okay, sometimes 8, like that it may be possible. Okay, due to some, this is also a fertilization error. Okay, so go to the next one. Polygyny. Note it on polygynous. Here, during oogenesis, after the meiosis, polar body 1 and 2 are not separated. In this condition, one male pronuclei, sorry, here the spell mistake, is fused to the 2 female pronuclei. This condition is called as polygyny. Polygyny means 2 gynecium used also united in a, which one, one male nuclei. That condition is called as, what is that? Polygyny. Okay, these are the errors during fertilization. See here, monogamy. Monogamy, what is the meaning? one male gamete fused with the one female gamete. This is the possibilities. Yes, it is. Always it happen like that. Polygyny, one male gamete fused with the many female gamete. That is called as polygyny. Polyandry. Polyandry means what is that? Sperm is different, but the unison is single egg only. Polyandry. Okay. Promiscuity. See here, the crisscross. Any male, any female, like that. These are the errors. Okay. So these are all no need. Just these three part is very, very important. We are all comes from the getting monogamy. One male, one female. Here, one male, but different female. Here, one male, different female, sorry, different male and one female. Okay. So there is a condition. See, just an example. Only, what is that? Honeybee is also an examples. These are all are, what is it? Polyandry. These are all are polyandry. See here, only one male here. There are so many female. Okay. So these are all are called as polygyny. Okay. See that next one. Polyandry. When two male pronuclei fuse with one female pronuclei, it is called as polyandry. Here male nuclei is poly. Andry means andresium. Take an example, male gamete. Two male gamete fuse with the one female pronuclei. As a result of formation of polyandry, that condition is called as, what is the polyandry condition? But in this category, embryo is dead. Note it down. This is very, very important. During polyandry condition, the embryo is alive. They may ask the question. The embryo is dead. That is very, very important. This is the another important. See here, marriage involves one woman and the more than one man. So this is a complicated one. Popular in certain Tibetan tribes and among the Inuit Eskimos, occurs in very poor culture where a number of working males are considered necessary to support one wife and children. Obey husband's wishes, one child from each husband. So these are all difficulties. Okay. Okay. So next questions. Sometimes they ask the question, what is that? The sperm produce substances called sperm lysine containing mainly which one of the following? Will you please? Hyaluronidase, hyaluronic acid, vitalinidase. Yeah. Go get Ajay.
Sir, the first one, sir.
Correct. First one is the answer. Okay. Hyaluronidase is the answer. Correct. Lysine. What is the next one? The preparation of the sperm before penetrating an ovum is called as, what is that? Capacitation, isn't it? Or insemination. Will you please? Which one is the correct one?
Sir, the second one, sir.
Shreya?
Yes, sir.
Okay. Anyhow, doubt is that, just check it once again, capacitation or inseminations. The preparation of sperm. Okay, this note it down. Tomorrow you'll tell because you are unsure. Okay. Fertilization is a chemical substance produced from. Fertilization is a chemical substances produced from a mature egg, acrosome, polar bodies, middle piece of the sperm. Fertilization. Okay, it's a chemical substance. Note it down the question. Tomorrow you tell me the answer. Today, the part 4 will be finished. We will pass on to the next one, 5th one. Do you have any doubt in this area? Ajay, Agil, and Jaswant.
No, sir.
Okay. Please follow it. Yeah. Okay. 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 We'll pass on to the next one is 5th part. See here. Lesson. So this is embryonic development after the fertilization. What happened inside? Before what we studied, male gamete, female gamete, gametogenesis, fusion of gametes. What are the changes which occurs? Okay. Then fertilization, then egg layer. Okay. So now last part have discussed the determination of sex. Okay, male gamete, female gamete, how it will be produced. That things also already discussed. Next, we will pass on to the embryonic development. Inside the embryo, how it will be developed. What are the changes which occurs within the ovum? You see here, all metazoans develop from a single cell stage zygote. Metazoan means middle. Okay, develop from a single cell stage zygote, isn't it? Male gamete, female gamete united together form as a single cell, isn't it? That's why all the human beings, okay, all arises from the single cell diploid stage zygote. Okay, the product of fertilization which divides mitotically after the formation of zygote, it will undergo the division is called as mitotically. Mitotically means, what is that meaning? Mitotically means body cell, isn't it? So zygotes also undergo mitotic division first, and through many sequential steps develops into the parent-like form. The process is called as embryogenesis. It included the cleavages. Okay, after the formation of zygote, it will not develop directly. It undergo the divisions. What are the divisions which occurs within the embryonic axis? That is called as embryogenesis. What are the changes? Here, blastulation, implantation, gastrulation, and organogenesis. Simply called as big O. First letter, you take an example, the big. Big means blastulation first, implantation second, gastrulation third, and organogenesis is the last one. B-I-G-O, big O. You have to, during the embryonic development, these are the cleavages occurs. Okay, we'll see one by one. See here. First, cleavage or segmentations. It is the mitotic cell division in zygote. With the following special features also happen. How? What are they? The cell division without growth phase. Note it down. This is very, very important. Cell division occurs without growth phase. Okay. Hence, the cell number increases, but size of the embryo remains the same as that of zygote. That condition is called as growth phase. The cell number increases. Okay, but the embryo will not increase their size. Okay, so that is very, very important. That phase is called as growth phase. This is the first. During cleavage, the size of the blastomeres keeps on decreasing as there is no growth of the blastomeres. Blastomeres, it will not grow. Note it on that point is also another one. And the zona pellucida remains intact throughout the cleavage division. Zona pellucida, it's the outermost layer, isn't it? Corona and zona. It will not, what is that happen? It is not cleavage. It present as it is intact. There is no activity throughout the cleavage. Inside only it happened. The outermost layer is present as it is. There is no changes. Okay, this is also a cleavage. During cleavage, there is no increasing in the mass of cytoplasm of the developing embryo. And next one is cytoplasm, there is no change. However, there is a marked increase in the DNA containing chromosomal material. So the nucleocytoplasmic ratio of the resultant cell after every division increases. Before that, what is that? There is no any, what is that? These are the changes. Mitotic division takes place, right? Okay, but growth phase is, what is that meaning? No, cell number increases, but size is not increases. Here also same. The cleavage of the size of the blastomeres keep on decreasing as there is no growth of the blastomere. There is no growth of the blastomere. That is simple. Zona pellucida remain intact throughout the cleavage. There is no changes there also, but during cleavage, what happened? The cytoplasm is also no increases. However, but DNA and DNA dependent molecules, what happened? Increases. This is the increasing. And then, so the nucleocytoplasmic ratio means what is the DNA as well as the cytoplasmic ratio of the resultant cell after every division increases. After that, what happened? It may increase. This is the first point, increasing order. Second one is O2 consumption is very high for the high rate of metabolism. Already in our report, during the development of the embryo, they need more energy, isn't it? So that time what happened? Automatically oxygen consumption will be need more. So the our what will happen? The oxygen consumption also, that is also breathing problem comes that time. O2 consumption is very high for the high rate of metabolism. Yearly few division in all blastomeres are synchronized except in human. This is also very, very important. So what are the two things is increases? Nucleocytoplasmic materials increases. O2 consumption is increases. Okay. Third one. In human, cleavage divisions are slowest in the animal kingdom. Animal kingdom, the cleavage always how it will be. It is not fast in manner, just like a multiplication of bacteria or virus. Okay. The cell division, it is a very slowest process in the animal kingdom. The rate of and the type of cleavage depends upon the amount of and the distribution of the yolk. What is the meaning of yolk? What is the main functions of the yolk? Already we discussed. They provide nutrition for what? Embryo. Okay, that is a very, very important. So depends upon the embryo only. Depends upon the yolk only. The cleavage will be very fast in manner. Okay, that is very, very important. Okay. Do you have any doubt in this part? I think very clear, I think, isn't it? Okay, go to the next one. Process of cleavage. What are the processes it may occur inside? It is a holoplastic equal type and the pattern is biradial. Biradial means, bi means what is that? Two. Radial means what is that? Radial aspect. Radial. Okay, it is not a longitudinal. It's a crosswise, isn't it? The interminate type. What is the meaning of divisions? See here, biradial and intermediate types. Divisions will be like this. The pattern, sometimes they ask the question. Cleavage of egg, biradial and interminate type. It takes place during the passage of zygote through the fallopian tube to uterus. Already I have sent the what is that? On AB, just you ask the ma'am, so we will get the idea. Okay. The first cleavage furrow completed after 30 hours of fertilization. After the fertilization, the first cleavage starting where? After the 30 hours. 30 hours means how many days? Only one day and quarter, isn't it? Quarter means 1/4. Okay. The 30 hours of fertilization appears at the animal vegetal axis, that is called as meridional first. Once again, the first cleavage furrow completed after the 30 hours of fertilization. Sometimes they are asked the question, 24 hours per day, add 6 hours. Okay. It divides the zygote completely into two blastomere. First division, how it will be? Two blastomere. One cell divided into two, that is holoblastic cleavage. This is also one of a holoblastic cleavage. Once again, biradial, interminate, whatever it may, interminate type. Okay, the first cleavage, 30 hours completed. Division, how it will be? The zygote completely into two blastomeres. Okay, so two blastomere, it gives what happened now? 4 to like that. See here, the second, this is the first division, and second division is also meridional, but at a right angle to first one. It is the complete earlier in one of the two blastomeres, resulting in a transient 3-celled stage. The second division, how many cell is present here? Transient 3-celled stage. The division is also there. Third division is equatorial, resulting into 8 blastomeres. It is slightly unequal. So see, this is the third division, 8 blastomere is formed. Here 3 cell stages there. First one, 2. Second one, 3. Fourth one, sorry, first one, 2. Second one, 3. Third one, what is that? 8 blastomeres. That 8 again, fourth division, you will get it there, 16. But it is unequal in nature. So it is again meridional, forming 16-celled stage called as morula. So already I asked the question, Ajay also answered it, isn't it? The morula, how many cell stage is there? 16 cell. This is very, very important. Okay, after that 16, what happened? It's also further divided into divide, you'll get 32 cell stages, isn't it? 32 cell stage. Okay, see, this is the divisions. See here, first one is egg, combination of both male as well as the female. As a result, the formation of zygote. After the zygote, what happened now? Undergo the cell division process. So we'll get the 2, isn't it? After that, 3 cell stage. Here they mentioned, but here they didn't give. Okay, it may be changed to 2 to 4 like that. Second or third, and again the 4 cell stage converted to morula, 16 cell. Blastula is a 32 cell stage. The inner cell is also. This is the developing stage or cleavage nature. Okay, this is very, very important. Significance of cleavage. Cleavage bring about the distribution of the cytoplasm of the zygote among the blastomere. Distributions, because once the karyogenesis as well as the cytogenesis take place, equal distribution is very, very important, isn't it? One cell is equal, another cell is unequal, means it is very complicated. It is very complicated one, isn't it? So that is the important. Cleavage bring about, what is that? The distribution of the cytoplasm of the zygote among the blastomeres. Okay. Increased mobility of the protoplasm, which facilitate morphogenetic movement necessary for cell differentiation, germ layer formation, and the formation of tissue and organ. This is the event. What is by cell differentiation? Each and every cell undergo the further development. These cell for brain, these cell for lungs, these are the cell for the growth of the kidney. Like that, they differentiated. After that, germ layer formation is there. And also finally, what happened? More than one cell united together forms a tissue, and more than one tissue forms a organ. Separate organ also formed. These are all are the cleavage significance. The restoration of the cell size and the nucleocytoplasm ratio characteristics of the species. Which one is decide their characteristics of the species? See here, cell size and nucleo or cytoplasmic ratio. That is very, very important. Which one is deciding the characteristics of the species? Already in our, but nucleus is very, very important, particularly DNA, RNA. For example, your character decided by your parental gene, DNA. Okay. Likewise, what is that? And also father and mother characters, genes also. But here, what is that? The species, for example, monkey, dog, whatever it may be. The combination is also there, no? So the DNA is a important one. Not only that, and cytoplasmic ratio, difference between nucleocytoplasmic ratio characteristics of the species. Okay. Unicellular zygote is converted into multicellular embryo. We are all multicellular. Already we discussed zygote is a unicellular, isn't it? The unicellular, how it will grow into multicellular embryo, mean due to the cleavage. That is very, very important. Okay. So after the cleavage, what happened on the embryo formation takes place. After the embryo formation, what happened when the cells are differentiated? After the cell differentiation, what happened? It forms as a tissue, and also the tissues are united together, form as a organ. finally entire organism formed. Okay. Okay. So this is the cleavage step. See that zygote. This is the single cell. Cleavaging of 8-cell stage. Which one is called as 8-cell stage? Morula, isn't it? After that, cleavage also undergo the blastola stage. This is all 32-cell stage and also 64 like that. And cross section of blastola, when you are going to see the inside, there is a plastocele is also present. Okay. So this is the another one. Gastrulations. Blastulation, blastula cross section, and gastrulation, and also finally see the structure of another one, blastomeres. So these are the cleavage, rapid cell division that lead to the multicellular embryo. We talk about single cell into multicellular embryo. Okay, these are all happen inside the mother cells. Okay, difference between ordinary mitosis. Okay, so this is also one of the mitosis process. Instead, single cell divided into two means it is also one of the mitotic division, isn't it? How it will be different from the ordinary mitosis? For example, our body is also containing mitosis cell division, isn't it? How it will be? See that cleavage result is decreasing of cell size, where normal mitosis the cell size will be decreases, whereas mitosis mainly maintain the cell size. Mitosis maintain the cell size. There is no cell decreasing order, isn't it? Here, result in decreasing of the cell size, whereas mitosis maintain the cell size. This is very, very important. Cleavage, note it down, very, very important. Cleavage, the size will be decreased, but normal mitosis, they maintain their cell size. First point. The ordinary mitosis, cell division is followed by a period of growth, while cleavage is not followed by the growth of the plastomeres. These are the another point. Okay, and there is a greater synthesis of DNA during cleavage. There is a greater synthesis in mitosis. Divisions are quick in cleavage. Divisions are quick in cleavage. Cleavage, what happened now? Quick compared to the mitosis. Consumption of oxygen is greater increase in cleavage. This is also another one. So consumption of oxygen is also high. Division are quick. DNA synthesis are quick. At the same time, whereas the mitosis, whereas the cleavage result, the size of this, it will be very less in nature. These are the 5 differences between the ordinary mitosis. Okay, then morula, then and these are the stages. Okay, we are going to plastulation and also gastrulation. That things it will comes. We'll see in the next classes. If you have any doubt, will you please ask me? Okay. First, what are the things already we studied? First of all, male gamete, female gamete, unison together. What are the meant by sperm structures, and then fusion of gamete, gametogenesis. Already that things also already we discussed past. Now, what we discussed, last class, what is that? Types of egg layer. That things also already studied. Here, what we are going to do? Embryogenesis. Embryogenesis related to these are the combination: plastulation, implantation, gastrulation, and organogenesis. That things we are studying deeply. Okay. Once again, just go through it because there are so many questions they were asked in this portion. Okay. So here you note it down. This is very, very important. Cleavage of segmentation is also one part, and then the oxygen consumption. Okay. Animal kingdom, the division, cleavage divisions are very slowest nature, but sometimes they told is a very fast in nature. Depends upon their yolk. Note it down. The cleavage depends upon their amount of distribution of the yolk. Take an example of ostrich egg. It contains which one compared to the normal egg in hen egg. How many times it more than that? 200 times, I think 200 to 300 times more than that of the normal egg. Ostrich is a big one, isn't it? Likewise. Okay. So that things also very, very important things. And then process of cleavage already told you biradial nature. How many minutes? 30 hours after the fertilization, the first cleavage started. Okay, zygote is a single cell converted into a multicellular organism that is called as embryo. Okay, so second division, how many cells is there? 3. Okay, and then third division, 8 cells is there. This is called as plastomeres. And then fourth, what is a 16 cell, that is called as morula. After that morula, what happened now? 32 cell and 32 to 64, that is called as blastomere, blastula, blastocyst, isn't it? That things also you have to. Significance already discussed. Cytoplasm of the zygote and among the blastomeres. Here what happened in a morphogenetic movement. Morphogenetic means cell differentiation, germ layer formation, and formation of the tissue and organ. Okay, and also it depends upon their ratio between the cytoplasmic nuclear substances, cytoplasm as well as the nucleo. That is called as karyogenesis and cytogenesis. Cytoplasm and nucleic activity, DNA and cytoplasm. And unicellular zygote converted to multicellular embryo. Okay, so how it will be? All those things: ectoderm, endoderm, blastocells, 8 cell stage, blastula. How many cells? 64 cell. I mean 32 and 64. Okay, these are the things. Finally, what happened? Multicell. This is the single cell converted to multicell like this. Okay, this is also a very, very important. That things already we discussed. I think today we will finish it up to this level. We'll go to the remaining things on the next classes. Okay, my dear students, if you have any doubt, please ask me. Okay, thank you, my dears. Thank you. Bye. Take care.
Thank you, sir.
Thank you, Guna. Thank you. Thank you. Thank you, ma'am.
Thank you,
Thank
sir.
you, sir.
Thank you. Thank you, all of you. Bye. Thank you.